Namaste, I am Miss Varsha Gaitonde and this video is about reverse breeding. One of the most important insights in breeding was heterosis, the observation that hybrid progeny typically are superior. Breeders can evaluate heterosis by controlled crosses of inbreds. Favorable allelic combination of the elite heterozygotes are lost in the next generation due to segregation of traits. Easy presentation of heterozygous genotypes is one of the greatest challenges in breeding. Apomixis has repeatedly been proposed as a way to preserve heterozygous phenotypes but has not yet led to breeding applications. A new technique called reverse breeding meets the challenge of fixation of complex heterozygotes, which was reported by Dirks in 2009, means 10 years back, and not has been commercialized yet. This was further made into a better form by Erickson in 2016. Reverse breeding is a novel breeding technique that makes use of genetic modifications to facilitate breeding of F1 hybrids by suppression of meiotic recombination to again produce F1. This starts with an elite heterozygous line and aims at the generation of homozygous parental lines. The subsequent crossing of these homozygous parental line produces F1 hybrid plants in which the original heterozygous line is reconstituted. Reverse breeding allows breeders to produce a hybrid in a much shorter time than with conventional technique. This novel breeding approach offers clear advantages over existing techniques due to the fact that in principle, any heterozygous plant can now be commercially exploited through resynthesis of suitable parental lines. This method is to produce parental line for the production of F1 hybrids to obtain F1 hybrids by crossing those parents developed through reverse breeding method. This invention further relates the DNA constructs used in this method. Coming to the concept of reverse breeding, it is nothing but the suppression of meiotic recombination during spore formation, production of double haploids of such spores, and crossing appropriate double haploid lines having desirable set of chromosome on the basis of matching molecular markers. Reverse breeding versus traditional breeding. In traditional breeding, superior heterozygous plants are generated by hybridization program, but in reverse breeding the engineered meiosis is developed and the parents or P1 and P2 are found, and again by crossing the P1 and P2 we can get a superior heterozygous plant just like traditional breeding. The homozygous lines are hybridized in order to reconstitute the original genetic composition of selected heterozygous plants is obtained in reverse breeding. Means we can get the genetic constitution of different parents by suppressing the meiosis. Objectives of reverse breeding. It provides alternative method for providing homozygous parental lines for the production of hybrids. To provide even more flexible in combining desirable parental lines for production of hybrids, to allow generation of chromosomal substitution lines that will facilitate breeding to study on an individual chromosome level. The reverse breeding technique is conceptually very easy but technically demanding. The need of reverse breeding. This is required because difficulty in maintaining hybrid stability. In order to get the parentage or exact constitution of the parental inbred lines, it is a good technique. To improve the hybrid performance, first the parental lines has to be improved. Inability to establish breeding lines for uncharacterized heterozygous, clonal propagation or apomixis preserves the parental genotypes but prevents its further improvement through adapting parental lines. According to the invention, it was surprisingly found that reverse of traditional breeding is possible. The steps in reverse breeding are providing a heterozygous starting organism, allowing the starting organism to produce haploid cells, creating homozygous organisms from the haploid cells thus obtained, then selecting the organism having desirable set of chromosomes, and finally reconstitute heterozygous by crossing homozygous organisms produced through this method and just check the original one and the newly produced one. The concept goes like this: by forward breeding, selection of the lines and production of hybrid plants. In reverse breeding, what we'll do, first we produce the lines, then select the heterozygous lines. That means selection of end product at the start of the breeding cycle is nothing but reverse breeding. As you know, in reverse breeding, what we'll do, we'll take a starting hybrid and we will make its haploid and we'll make the diploid condition or double haploid condition. Here different organisms or different progenies are obtained, but we will take the only lines which have the original parental constitution, or in other words, if we'll cross the double haploid lines which are having the same constitution as parental lines are only selected. This is how the reverse breeding of meiosis takes place. By normal meiosis, there will be genetic recombination, but in reverse breeding, the meiosis is inhibited so that only the haploids are obtained, and by the double haploid production, the double haploid conditions which give it won't allow the recombination here. Likewise, the chromosomal substitution lines are also constituted by reverse breeding. Per chromosome breeding is also possible, means we can target a specific chromosome. We can obtain a desired set of chromosome. It can be one of a number of variants. In the case of original starting hybrid is to be produced, the homozygous organism produced according to the inversion should together have the exact set of chromosomes. This is achieved when both parents have the same set of chromosome as the gamete form the hybrid. It is also possible that the new material line has only some of the chromosomes of the original maternal gamete in the background of the original parental gamete. That is nothing but chromosomal substitution. It is also possible to combine the new lines which has one or more but not all the chromosome of the original parent with a different parent in breeding. The working principle of reverse breeding is, first one, selection of homozygous through reverse breeding is the main motto. Here what we will do, we'll take a hybrid, or by forward breeding we'll make a hybrid by crossing parent 1 and 2. That hybrid further by reverse breeding, what we will do, we'll go for conventional breeding. And when we go for conventional breeding, it is very difficult to find out the original parents because of the recombination and certain mutations, whereas in case of reverse breeding by RNAi technology or by suppression of meiosis, we can get the end product as same as the original parent. Suppression of meiotic recombination include two steps. One is to produce gamete from heterozygote, and the second is suppression of recombination during spore formation. In the first process, suppressing gene required for meiotic recombination, and in the second step, complete knockout of the gene by RNAi to knock down the formation of DMC1 homolog to Rec-A protein, which is a meiotic specific recombinase essential for the formation of crossover. The exogenous application of chemical compounds that cause inhibition of recombination during meiosis would speed up the application of reverse breeding. Example, mirin. It's a chemical compound applied externally. Genes responsible for meiotic recombination are DMC1, REC-A, and SPO11 gene. These are involved in RNAi knocks down the function of these genes during spore formation. Exogenous application of chemical compounds that cause inhibition of recombination is mirin, as I have already told. Mirin causes G2 or G2 phase, that is gap2 phase arrest, and inhibits the phosphorylation of ATM, that is ataxia telangiectasia mutated is equal to serine or threonine protein kinase. That means the meiotic inhibition is done either through the genes already there or by application of the different chemical compounds exogenously. So far we have seen the genes involved in this process. Now the reverse breeding relies on achiasmatic meiosis. The first and foremost point is on the function of crossover. In flowering plants, the formation of crossover during meiotic prophase relies on synaptonymal complex. During the crossing over, two homologous become physically joined when the distal ends of one chromatid is attached to the proximal end of a non-sister chromatid and vice versa. The resulting of joined homologs is called as a bivalent. Production of double haploid is another type. Here, tissue culture of immature pollen should be done using tissue culture techniques referred as anther culture or pollen culture anything, and isolated microspore culture where immature pollen grains grow to produce colonies of cells. The colonies are transferred to media with different plant growth regulators and sugars to induce growth of shoot and then roots. The third step is selection of complementary line or parents through marker-assisted selection. Through marker-assisted selection, the codominant markers or the fragments are easily differentiated, and after that crossing appropriate double haploid lines on the basis of matching molecular markers to develop superior hybrids. This will give the similar complexity between the original and the newly made hybrids. The comparison of parents and the original parents and the hybrids parents will give a clear-cut results of reverse breeding. A chiasmatic chromosome remains as univalent. Chiasma, that is bivalent, promotes segregation of homologs to opposite pole, that is regular disjunction. In the absence of univalence, and the homologs may segregate to the same pole instead, that is non-disjunction. This leads to unbalanced chromosome number in the spores. Consequently, achiasmatic plants are highly sterile. More univalents are present, the more aneuploid pollen are formed. Chances of finding the complementing parents. Maximum number of different double haploids obtained from a heterozygous diploid in reverse breeding experiment equals to 2 raised to x, with x being the basic chromosome number. The probability that two double haploids form a pair of complementary parents equal to 2 raised to x by 2 raised to x whole square, that is equal to half raised to x. Probability that they upon crosses do not reconstruct the original genotype is 1 minus half raised to x, that is equal to 2 raised to x minus 1 divided by 2 raised to x. The number of combinations between different double haploids, presuming that reciprocal crosses result in the same phenotype, is n into n minus 1 by 2. The probability of at least one complementary combination of two double haploids from n double haploids is given as 2 raised to x minus 1 divided by 2 raised to x whole raised to n into n minus 1 by 2. The chances of finding complementing parents is given in short form, where the first step, probability that two double haploids form a pair of complementary parents equal to half raised to x. Then the probability that a complementary double haploid combination is absent is 2 raised to x minus 1 divided by 2 raised to x, as I described in the previous slide. Presuming that reciprocal crosses resulting in the same phenotype is n into n minus 1 by 2. By these two, we can conclude that the probability of at least one complementary combination of the two double haploids from n double haploids is 2 raised to x minus 1 divided by 2 raised to x whole raised to n into n minus 1 by 2. The technical realization of reverse breeding. The factors governing crossover frequencies, internal factors like genetic background, morphological and developmental differences, then external factors like environmental influence and chemical treatments, physiological stress like temperature shock or UV exposure. In recent years, many proteins are involved in crossover formation have been identified. Example, in tomato, overexpression of MUT-L homologs, that is MLH1, lead to 10% increase in chiasma frequency. In Arabidopsis, a twofold increase of recombination frequency upon the crossover of radiation sensitive phi1, that is RAD5. Identification of target gene in reverse breeding. A gene residing within the genome of an organism which upon qualitative or quantitative modification of its expression results in a modified meiotic process. To know their functional homologs from other species, genes that are involved in meiotic recombination constitute potential targets for modification in which meiotic recombination is to be suppressed. In fact, they encode perform the same or a similar biological function. Knockdown of modification of target gene. This is done by preventing transcription where RNA oligonucleotides, DNA oligonucleotides or RNAi molecules against the target gene promoters. Destabilizing the target gene mRNA. Here, antisense RNA, RNAi molecule, viral induced gene silencing molecules, co-suppressor molecules, RNA oligonucleotides or DNA oligonucleotides are utilized. Inhibiting the target gene expression product. In this, dominant negative nucleic acid constructs overexpression of one or more suppressors which interact with the target gene product. One or more chemical compounds are used. Target gene may be silenced by silencing molecules derived by graft transmission. The exogenous application of mirin as an inhibitor for the MRS11 RAD50 NBS1 complex that cause inhibition of meiotic recombination. Chemical compounds are ethyl methane sulfonate, then nitrosomethylurea, hydroxylamine proflavin, and methyl N-nitrosoguanidine, and ethyl N-nitrosoguanidine, so on. The introduction of one or more mutations into the target gene leading to perturbation of its biological function. Physical means that can be used to comprise UV radiation, fast neutron exposure, X-ray and gamma radiations can also be used for the chromosome or crossover suppression. Genetic element is a transposon, DNA or retroviral elements can also be used. Coming to the effective suppression of recombination process. For this, the reverse breeding relies on effective suppression of meiotic crossover. Example, in Arabidopsis, ASY1 in rice, ASI1 homolog is to pair 2, the mutant of which display univalence at end. Other mutants with similar phenotypes are DMC1, SDS, PTD and SPO11. The knockdown of gene expression essential for reverse breeding can be achieved by targeting genes using various methods. Exogenous application of compounds that cause inhibition or omission of recombination during meiosis would speed up the application of reverse breeding enormously. Coming to RNA interference. It is an evolutionarily high conserved process of post-transcriptional gene silencing by which double-stranded RNA causes sequence-specific degradation of mRNA sequence, which was first discovered in 1998 by Andrew Fire and Craig Mello in the nematode worm C. elegans and later found in the wide variety of organisms including mammals. This is how the RNA interference works, where the homologous synapse ASY1 or SDS is involved, and SPO11-1 has double strand break formation and DMC1 has strand exchange. This is the early and middle prophase target genes which work normally and helps in the crossover and normal functioning of meiosis. When RNA interference works, then the promoter region or terminator and the whole promoter region is programmed. Here the DNA construct integrated in the plant genome. Pre-RNA is constructed. From there mRNA is synthesized and that will fold back, and RNA interference will lead to non-functioning of that, means the complete functioning will be impaired. This is how the RNAi works. There are different kinds of RNA interference like miRNA, siRNA and shRNA. In that, RISC complex, dicer, argonauts and all these things are involved in inactivation or silencing of the mRNA. Coming to viral induced gene silencing. This is a technique used for silencing the expression of specific endogenous genes in a plant. VIRUS DNA vector is usually introduced into the plant using agrobacterium or specific plant virus. Viral vectors are composed of a modified viral genomes and include a fragment from the host plant gene to be silenced. It is based on a plant defense mechanism that limits the severity of natural viral infection in plants. It avoids the need for time-consuming plant transformation and regeneration process. Different viral vectors are for TMV, is a vector, species is Nicotiana bentianum, and symptoms are severe. Efficiency is fair, better with antisense, improved with inclusion of direct inverted repeats. Likewise, PVX, TGMV, TRV, SVISS, all these are acting as viral vectors in different organisms. The VIRUS mechanism goes like this: random cDNA library from where the clonal fragments are taken as a vector, then candidate gene is identified and virus binary vector is constructed. Through agrobacterium tumefacian transformation, it will be injected into the plants by toothpick, vacuum or anything, or syringe. Then viral spread and silencing occurs. After 2 to 3 weeks, the plant phenotype will be entirely changed. Coming to the applications of reverse breeding. This is used in transfer of CMS, used to produce a maintainer line from homozygous cms-line, that is, production of B line or maintainer line from A line or male sterile line. Reverse breeding provides seed propagated varieties in species that now are commercially multiplied by vegetative propagation technique. The technique is used to improve seed production in hybrid seeds or hybrid crops, reconstitution of heterozygous germplasm, breeding on a single chromosome level is also possible here. Reverse breeding and marker assisted breeding. High throughput genotyping speeds up the process of identification of complementing parents in population of double haploids. It helps in the study of gene interaction in heterozygous inbred families. The sequencing of population that segregate for trait on a single chromosome allow the quick identification of QTLs when genotyping is combined. It aids in generating chromosome-specific linkage map. Fine mapping is also possible for genes and alleles and helps in studying nature of heterotic studies. The first and foremost is reconstruction of heterozygote. It is the application part. Reconstruction of heterozygous for crops where an extensive collection of breeding lines is still lacking. Reverse breeding accelerates the development of varieties. In crops, superior heterozygous plants can be propagated without prior knowledge of their genetic constitution. Breeding at single chromosome level. Many interesting characteristics in plants are based on polygenic gene interactions, very often located on different chromosomes. These quantitative traits are therefore not easy to breed on. Reverse breeding explains how chromosomal substitution lines can be obtained when reverse breeding is applied to the F1 hybrid of noun parents. These homozygous chromosome substitution lines provide novel tools for the study of gene interactions. When crossed with one of the original parents, hybrids can be formed in which one of the chromosome is homozygous, whereas it is also possible to produce hybrid in which just one chromosome is heterozygous. The former allows the study of epistatic interaction between the background and genes contributed by substitution chromosomes. Offspring of plant in which just one chromosome is heterozygous will segregate for traits present on the chromosome only. Selfing plants that carry a substituted chromosome will allow breeders to fine-tune interesting characteristics on a single chromosome cell. This could bring improvement in the inbred breeding line carrying introgressed traits. Transferring the CMS. In order to convert a desirable inbred line or pure line into similar line but with CMS background, reverse breeding is very helpful. The use of large number of nuclear genetic markers so that difference between the chromosomes of CMS and the fertile donor can be more easily determined. CMS donor is made homozygous that confers recombination suppression. First cross is now made by pollination of the CMS homozygous of interest. Homozygous recombination suppressed line with pollen of desired line. Resulting F1 progeny will contain CMS and 50% of the chromosome of desired line. In the meiosis of resulting F1 plants, no recombination occur as a result of inversion. This again fertilized by pollen of desired line, and the resulting seed is genetically identical for the nuclear gene to the original desired line, but now has acquired the CMS plasma. That means the plasma genes are only transferred. So the second cross with the desired line, seeds have acquired the CMS plasma of donor line. In the newly achieved CMS or nuclear composition, no transgenic gene or plants have to be retained due to segregation of the transgenic locus that is responsible for suppression of meiotic recombination, and because the transgenic plants are not retained. Here how it goes, the transfer of CMS line. This is the entire procedure. Coming to the wide seed propagated varieties in species that now are commercially multiplied by vegetative propagation technique. The logistics of producing plants through vegetative propagation is much more difficult than through seeds. Seeds can be stored easily and often without problem for a long time. In the case of vegetatively propagated material, it is much more difficult to respond to varying commercial needs for new planting stock. Vegetative production is labor and technology intensive and thus relatively expensive. Diseases, especially viruses, are a constant threat to vegetative multiplication. Therefore, many Viruses are not transmitted by seeds but are easily transmitted to clonal offsprings obtained by vegetative propagation. That's why some countries have strict quarantine regulations governing the importance of vegetatively producing plants. In addition, not all genotypes perform equally well in vegetative propagation. Though reverse breeding, the genotype of heterozygous clonally propagated plant can now be resynthesized and hybrid seeds with the said genotype provided. Using reverse breeding to improve seed production in hybrid seeds. Difficulties like poor seed production capacity of the maternal line of the hybrid, or a difference in flowering time, crop height, or flower morphology of the maternal and paternal lines of the hybrids. This hybrid can be resynthesized using lines which differ from the original maternal or paternal lines. By selecting a combination of lines which allows the production of commercial seeds of both high quality and quantity, the commercialization of hybrid becomes economically feasible or more effective and attractive. Coming to comparing the end product of reverse breeding and conventionally bred crop. The end product of reverse breeding will be similar to parental lines obtained by conventional breeding. The RNA interference silencing is restricted only to meiotic crossover suppression, but there will be no change in the DNA sequences of reverse bred plants. Thus, resulting offspring can be regarded as non-genetically modified. Coming to the difference between marker-assisted reverse breeding and RNAi-mediated reverse breeding. In marker-assisted reverse breeding, no need of gene silencing, 1 to 1.5 years for the development of homozygous line, no limitation in crops with less than 12 haploid chromosome number, and not limited for crops where double haploidy is not possible. Whereas RNAi or RNA interference mediated reverse breeding, there is a need of silencing. It takes 2 to 2.5 years for the development of homozygous lines, limitation in crops with less than 12 haploid chromosome numbers, limited for crops where double haploidy is not possible. It's a young technique, hence requires more research to suppress crossover. Consequences of reverse breeding. First and foremost is consequence for food and environment safety. RNA-directed DNA methylation transmitted to the offspring will only have an effect on meiotic recombination and no genetic modification related DNA sequences. Reverse bred crops are similar to those of parental lines and F1 hybrids obtained by conventional breeding, so it is very safe. Effect on environment. Because of the parental lines produced by reverse breeding and the subsequent produced F1 hybrids do not contain any genetic modification related DNA sequence, and because a possible RNA directed DNA methylation, that is the consequences for the environment, are in principle similar to those of parental lines and F1 hybrids obtained by conventional breeding. Such conventionally bred parental lines and F1 hybrids can serve as baseline. Effect on food and feed safety. The F1 hybrids are usually used as food and feed production. Therefore, these are considered as the consequence for food and feed safety rather than the parental lines, which are the actual end products of reverse breeding. F1 hybrids obtained by crossing of reverse breeding derived parental lines do not contain any genetic modification related DNA sequence, and a possible RNA directed DNA methylation that is transmitted to the offspring will only have an effect on meiotic recombination. Coming to the limitations of reverse breeding. Development of reverse breeding is limited to those crops where double haploid technology is common practice. Example: cucumber, onion, broccoli, sugar beet, maize, pea, sorghum, etc. There are some exceptions like soybean, cotton, lettuce, tomato, where double haploid plants are rarely formed or not available at all. The technique is limited to crops with haploid chromosome number of 12 or less, in which spores can be regenerated into double haploids. Moreover, it's a costly affair. The main application of reverse breeding is when the private companies or any of the person wants to know the parentage of any ruling hybrid in the market, or when the particular plant is patented or registered in PPVFR Act and a person or researcher wants to know its exact parentage to use it in further research, it is a best technique. However, it is a theft in the research or scientific society, but this somehow help for the future breeding programs. Summarizing the reverse breeding concept: first, the selection of plant at heterozygous level, then suppression of the meiotic recombination, by which spores or gametes containing random combinations of maternally or paternally inherited chromosomes are obtained, by regulation or regeneration of haploid or double haploid plants. Diploid lines containing random combinations of maternally or paternally inherited chromosomes are obtained. Identifying desired set of double haploid plants helps in crossings of complementary lines. That is the complete picture of reverse breeding. Genes with proven record. End product is a traditional F1 hybrid. Traditional seed production. Chromosomal substitution is a bonus here. Then breeding or recombination per chromosome is manipulated. Thank you. I hope you liked my video, and do not forget to subscribe.